All right. If we can come on back together for our last session. Our first speaker this last session is Çiğdem from OHSU. She's going to be talking to us about spatial heterogeneity, sample size effects, and prostate cancer.
Yes, thank you so much. My name is Çiğdem. I'm a postdoc at Cancer-Early Detection Advanced Research Center at OHSU. And I'm a mathematician by training, and I develop scalable and inherently interpretable algorithms for understanding single-cell biology, mostly multiomics. During my PhD, I developed spatial temporal models for infectious diseases, and I also applied to other spatial temporal data. But today, it will be mostly about how I applied spatial statistics to spatial data, like the very first talk also mentioned about Aaron's talk was showing how GIS tools were applied to spatial data. Hence, that's why the map on the cover, but also, of course, neighborhood analysis will be included as well. But today, I'm very excited to share our recent work on spatial analysis of cancer, prostate cancer, especially androgen receptors' role across multiple scales. Prostate cancer is the second lead cause of cancer death in men, and prostate tumors are typically graded using the Gleason score, which classifies cancers into 5 different categories, and it's done by looking at the H&E images. You have seen this slide before from Jonathan's presentation the first day as well. And he also mentioned that it's very difficult to differentiate, especially intermediate risk tumors, especially Gleason 3 and Gleason 4s. Even though, also he mentioned, even though most prostate cancers are localized at the time of diagnosis, many low-risk tumors are overtreated or removed, which decreases the overall life quality of the patient. And many high-risk tumors are diagnosed as indolent, and that also, of course, decreases the survival. So this highlights the need for more accurate methods to differentiate cancer subtypes. We cannot just use this whole tissue to understand disease subtypes. We have to go into more detail. So this is when multiplex imaging comes into play. We are looking at multiplex imaging of a prostate cancer tissue. We can see distinct cell types, molecular heterogeneity, such details that traditional H&E staining cannot provide. So this ability of multiplex imaging reveal more specific information, gives us deeper insights into tumor biology, and we are going to use this to enhance our ability to classify patients and tailor treatments more effectively. So we generated a spatial single-cell data set using cyclic immunofluorescence, CYSIF, from treatment-naive localized tumors across 3 groups. Tumor-adjacent normal, Gleason 3, and Gleason 4. The data set includes whole slide images ranging from 2 to 20 millimeter. After staining, imaging, and quenching tissue sections for 10 rounds, we obtain approximately 40 markers from a single tissue section. And using AI-based segmentation algorithms, we obtained almost 700,000 cells, which were classified into 27 cell types using multi-step clustering approaches. We identified 10 distinct cellular neighborhoods using these cell types and their location across all clinical groups. We have so many results on this data. In this presentation, I will focus on spatial analysis of androgen receptor, AR, in prostate cancer, especially in the stroma at different biological scales. Ina also showed different analysis at different scales. I will do a similar analysis here. And this will provide us a multidimensional view of AR's role in prostate cancer progression, molecular, cellular, and tissue level. And so the reason that we are going to focus, because even though AR is well known for its role in cancer progression, it's also expressed in stromal cells, where it plays a major role. In this diagram on the left, you can see an stromal cell that expresses AR, and it by releasing chemoattractants, growth factors, also controlling the ECM extracellular matrix production and controlling the immune response, it creates a habitable environment for the tumor cell. And it has been shown that the loss of AR expression in the stroma linked to cancer relapse and poor diagnosis. However, there are no previous reports of delineating non-immune stromal cell types in human adenocarcinoma based on AR expression. So our hypothesis is that spatial analysis of AR-positive stromal cells will help us understand the disease better. And that's why we investigated prostate cancer progression with the following questions. So at the cellular level, which stromal cell types express AR, and what is their spatial interactions? How do AR-positive non-immune stromal cells drive disease progression? And specifically at the molecular level, how combined expression of AR with other epigenetic markers impact survival? So as our first analysis at molecular level investigation of AR, we examined the combinatorial expression of epithelial markers in single cells and found significant differences in the expression of AR, AMACAR, and two other histone markers across all clinical groups, and specifically much higher in Gleason 4 high-risk tumors. When we look at CK8 positive luminal cells, this combined expression did not show any differences across clinical groups. And we will look at this observation at multiplex imaging. You can see in the H&E image here, two glands. One is annotated as tumor adjacent normal. The other one is Gleason 3 gland. And you can see the AR and AMACAR combined expression in these tumor glands. And you can see CK5 and 8 as well. And CK5 is basal cells are around this gland. So, and then it's lost here. So it's a tumor gland now. And then you can see the histone marker expressions on the left. And there's no illumination for those in tumor adjacent normal gland, whereas like there's slightly expression of those markers in Gleason 3 tumor. And then when we look at the an example of a Gleason 4 tumor, we see that again, AR and AMACAR here, and CK5 and 8 mostly lost, and then histone markers combined with IR and AMACAR very obvious expressed together. So next, we investigated these markers, spatial patterns at the molecular level using global spatial autocorrelation. That's from spatial statistics and existent GIS as well. It helps identify clustering patterns, whether nearby locations are similar, that gives us positive spatial autocorrelation, or nearby locations are dissimilar, like a checker box pattern, high expression of a marker in a cell. If they avoid each other, that looks like this, and it gives us negative spatial autocorrelation value. And of course, we used Moran's eye to calculate this. And using Moran's eye, we compared our distribution to random distribution to see if the observed pattern is statistically significant or not. So I calculated the Moran's eye for all the epithelial markers, but especially to look at the markers that we saw that the combined expression was differentiative of the clinical groups. We see that certain markers show higher clustering compared to others, such as AMACAR showed high clustering. And the thing is, we did not see any spatial pattern differences across clinical groups. For the cell level analysis, we identified 13 myeloid and lymphoid cell types and focused on mast cells because they were very prevalent across all of the clinical groups, almost 20% of the immune cell types. So we discovered 2 distinct mast cells based on their gene expression, protein expression, sorry, and we decided to investigate their spatial patterns. So we analyzed spatial interactions of mast cells based on AR expression using the interaction matrix, which was presented before as well in this conference, which is a descriptive statistic of spatial graph. It quantifies the number of edges that node belonging to mast cells share with other cell types. Our findings show that AR-positive mast cells interact closely with regulatory T cells in prostate tumors, and in addition, M2 macrophages in Gleason 3 tumors, and no spatial interaction in tumor-adjacent normal tissues. We investigated cell interactions using co-occurrence probability as well, which measures how likely cell type A is to appear near cell type B compared to its overall likelihood at a given distance. So this is a function of distance. We found AR-positive mast cells have a spatial association with M2 macrophages. This validates our result from the previous slide. This is a G3 patient. And we also assess the clustering of mast cells, whether they express AR or not, using Ripley's L function. This function is similar to Moran's I, but for discrete values such as cell types, which determines if cells are randomly distributed, dispersed, or clustered at different scales. We found that AR-positive and negative mast cells are not spatially clustered, but actually spatially dispersed. Finally, at the tissue level, with neighborhood analysis that I all learned about from the papers from Nolan Lab, we identified co-localized groups of cells that are commonly co-occurring in our data set. We identified 10 recurrent neighborhoods in tumor stroma based on 27 cell types. Top 4 neighborhoods are defined by an increased occurrence of AR-positive stromal cells. The bottom 6 neighborhoods are defined by a lack of AR-positive stromal cell occurrence. And on the right, we compared the recurrent cellular neighborhood frequencies across all clinical groups, and our results show that AR-positive stromal neighborhoods distinguish tumor-adjacent normal tissues from prostate tumors. To focus effects of spatial variability and sample size, we created in silico tissues tiling whole slide images. Dennis also mentioned about this earlier. After dividing each tumor sample into multiple regions, then performed, and also we sampled from these tiles in 2 different independent experiments, 10 in silico tissues, tiles from each grade clinical group, and also 50. And we show that the same neighborhoods were identified in both experiments, and AR-positive stromal neighborhoods frequency was again found to be significantly different in tumor-adjacent normal versus Gleason and Gleason 4 tumors. And to further show the robustness of these results, we performed subsampling and leave one out analysis as well. So for 100 iterations with different seeds, we subsampled the data, 50% of the data, and performed neighborhood analysis and compared tumor-adjacent normal versus the tumors. Again, the same if the AR-positive stromal neighborhoods differentiate or not. The experiments fall below this red line, which is p-value 0.05, means that our claim was true at the 86% and 90% of the time overall. And then for the exclusion analysis, this allowed us to assess the impact of patient-to-patient variability on the results. So here, each that is an experiment where a patient was dropped, and we run the neighborhood analysis and compared across clinical groups. So I'm circling back to molecular level analysis with my last slide. To better understand biological targets in AR positive stromal cells, we moved from imaging to gene network analysis using single cell ATAC sequencing data from the same patient's tumors. We identified regions with increased chromatin accessibility in AR positive myeloid and lymphoid and also non-immune stromal cells. These regions were significantly enriched for the genes that are regulating NF-kappa B signaling. So we also run a gene activity analysis on the same data set, and we saw that AR positive stromal cells showed enhanced activity in NF-kappa B gene, and also in addition, hypoxia. We validated HIF1 alpha and NF-kappa B signaling in these AR positive stromal cells using multiplex imaging on additional patient samples. So hopefully I convinced that AR has a big role in how we can understand the tumor biology and its progression, and can help us better subtype disease, find the subtypes of the disease. And we showed AR's role in spatial interactions between cancer and stromal cells. With that, this is the summary result of our, summary figure of our results. And I thank all my co-authors on this paper. The QR code goes to the paper, and also CEDAR for funding this research.
Thank you. Time for a question?
So I know that there is a lot of RNA-based imaging out there. Have you tried any of the fusion technologies to take the information from the RNA to overlay it onto these neighborhoods to see if you can see some of the networks that are underlying your epigenetics?
That's a great question. So we found a similar single cell RNA, not spatial, in the literature, and we found exactly AR positive stroma expresses NF-kappa B signaling. And now currently we are working on site-seq type of like protein and RNA expression on this tissues spatially. Currently, the data is not here yet, but we are currently working on it. But the existing single cell RNA experiments on similar clinical grades confirmed our AR positive stroma and NF-kappa B signaling.
I have one more question. So the AR positivity in all these different kind of non-prostate cell lineages is kind of, I mean, it's surprising for me. How much of it, you know, because I haven't dug into the paper, how much of it do you think is it really endogenous expression, or is it like kind of been extracted out of the images and is it a reflection of like how the cells are interacting with the tumor?
Yeah. That's a great question. I'm not a biologist again, but from the analysis, I can tell more about that. So we definitely looked at, like, we identified non-immune stromal cells and their AR expression, and we definitely saw that those number of cells, AR positive stromal cells, decreases with higher risk tumors. That's, and then we wanted to delineate how they interact with each other. So, yeah, I think it's not just the AR and the just imaging, but we identified cell types, and one of the talk was yesterday. They talked about cell types, granular cell types doesn't make maybe much sense. We shouldn't maybe trust. We should look at the functional role. So this was kind of a little bit example of that. So I think, I believe that it's the AR's role in the cells rather than overall AR expression.
All right, thank you.
Thank you.
Our next student is actually from my lab. Bryan Cannon is going to be telling us a little bit about multimodal analysis of human microglial states and how they behave in situ in the human CNS and Alzheimer's disease.
Hi, so like Sean said, I'm in his lab. I'm a PhD student. Today I'm going to talk to you about multimodal mapping of activated microglia in health and Alzheimer's disease. So microglia, if you don't know, are the brain's resident immune cell, most akin to macrophages. They have lots of roles in neurodevelopment, disease, injury, and neurodegeneration. So we've been able to see them in tissue, particularly post-mortem, forever, but actually being able to analyze them has been very difficult. Most of the time, you don't get live human tissue, so you have to look at the cells after a patient has passed. Mouse is easier, but the phenotypes don't necessarily reflect what's going on in human. So we want to be able to characterize and look at human microglia, which the only way to really do that and establish a reliable, robust rubric of this is using some of these newer multiplex and spatial technologies. So how we've done that in our lab is by first gathering different human microphenotypes across different brain regions. And so we started up with a single patient where we collected 5 different brain regions. And I particularly want you to focus on the hippocampus and the caudate nucleus on the left. Hippocampus is a brain region primarily involved in memory consolidation. Caudate is involved with general signal integration. And so we took these FFP fixed formalin samples, stained them with a panel of around 40 or so antibodies. And those antibodies primarily captured a bunch of different brain structures, brain cells. And in that dark blue little box there, we have our microglia and myeloid cell phenotypic markers, because that's, again, the population that we really want to characterize here at the spatial and proteomic level. So we then image these samples using Mibitov, and we get these beautiful images that we can tile together and show the integration of all these different proteins. And so this is the human hippocampus, and this is showing different structures. In the red, we see different neuronal structures, green synaptic structures. In the blue are different non-microglial, glial proteins, and then nuclear and vasculature structures. But of course, we're all here for the microglia. So we have our typical microglia structural markers, IBA1 and CD45. And the way we go about actually getting those cells out with their aberrant, weird shaped, non-nice circular processes is using a tool I developed in the lab called Easy Segmenter. It uses relatively simple image masking algorithms, but does so in a fast and efficient way so that we can grab all these microglia and turn them into the single cells we know and love. And then once we have all that data, we can look at how the microglia densities look in the different brain regions. And just in the left, you can see how the microglia tend to be a lot more prevalent in hippocampus compared to cerebellum, which is a different brain region involved in fine motor control. And then we can also break that down by more functional brain type here on the right, gray and white matter. So now that we have these cell types, now that we've kind of looked at a high overview of where they are, we want to be able to, again, phenotype these in a way that makes sense to us as humans. So the way we normally go about that is by clustering. And when we run a standard Flowsum clustering process, we see that, sure, we're able to get clusters, but they're all kind of meshed together. We don't really get nice separation. So what we instead tried to do is we took all of our different proteins and ran them through a trajectory-based algorithm, in this case, Scorpius, and mapped out how each of these cells and each of these phenotyping proteins align along a more continuous state, representing differential functions. So then if we take where each of those microglia map within that continuum and map it back over the original UMAP projection, we can see that this mirrors a much nicer continuous flow of these microglia cell states. And that with this continuum, we can also then break down how the microglia densities look within these different brain regions and see that this continuum varies with anatomical location. So this microglia state continuum curve can also be broken down at the individual protein contribution level. And so we can use these curves. In this case, it's between different brain regions, as you'll see in a little bit, between health and disease, to statistically to find statistical significance between differences in these microglia phenotypes by just looking at the differences of these curves between these different regions or disease conditions and so forth. So now that we've established this framework on a single patient of being able to capture all these different microglia phenotypes in a continuous manner, we wanted to expand this out to a much higher sample size and look across multiple human brains. So we have 42 additional samples, primarily focused on as I mentioned before, the hippocampus and the caudate nucleus, broken down between Alzheimer's disease and healthy human brains. And overall, that data set resulted in 72,000 images. This is all heavily downscaled, but we then captured 40,000 microglia from all these human brain samples. So going back to, we established a microglia state continuum for this new cohort of human brain samples. And again, everything that we're going to see here is essentially measuring curve differences, in this case, between these two different brain regions, the caudate and the hippocampus, in healthy patients. And so what we're seeing here is between these different regions of the spectrum, the low to the medium to high, is anything in the blue are proteins that were enriched in microglia in the caudate region, and anything in this orangish salmon color are proteins that are particularly enriched in microglia within the hippocampus region. And so there's some consistent immune phenotyping proteins that we see across the entire spectrum. In this case, ApoE, which is a signaling protein and primarily enriched in the caudate, P2RY12 and HLA-DR, more typically assumed with immune activation in the hippocampus. And then, of course, we want to actually be able to look at the actual image data and visually interpret this. And again, these are the primary ones, though there's additional markers as well. And this framework kind of allows us to be able to look at both the phenotyping as well as different functional markers that we had in our panel and how those differ across these different microglia phenotypes. So now that we've established that we can look at microglia and phenotype them in a continuous manner at the proteomic scale, we wanted to bring in and check our work using a different modality, in this case, single-cell attack-seq data that we had obtained from REM courses. And so we pulled out just as microglia and then clustered it into 3 different bins, in this case, again, low, medium, and high. And then when we break down the actual gene contributions to these different clusters, on the right side of this heat map, you can see that all of our MSC associated protein, or MSC, the proteins that we were looking at the MSC earlier, all those associated genes fall into this high cluster, which is also interesting. And that we had genes that fell into this more epigenetic MSC low cluster were primarily focused around neuronal support and immune resolution genes. And the interesting part about this is essentially what we're seeing is that a lot of the MSC proteins that we looked at in the MIBI data set that we showed earlier were more than likely pulling out more nuanced immune phenotypes that we can get because we're looking at single cell, we're looking at fine detail, where when we kind of pull back to the high level, we can still see the general direction that of low to high that we saw in the MIBI data. But in this case, it's just at a broader scale, and it kind of gives us some insight into what might be going on at the other end of the spectrum, in this case, the support and resolution genes. And that's backed up when we run a gene ontology enrichment exercise, where we see in the epi-MSC high cluster, we see these immune activated gene ontology programs, representing, again, a wide variety of programs that we can interrogate more deeply with spatial proteomics and MIBI. And then on the other side, in the epi-MSC low clusters, we see, again, these different pathways that are particularly reflective of microglia support of synapses. So now that, again, we've been able to establish both a proteomic and a genetic-based multimodal mapping of these microglia cell states, we want to see, okay, how does this actually look in disease? In this case, we're going to look at Alzheimer's disease, as I mentioned earlier. So again, using that difference in curve MSC statistical comparison that I mentioned earlier, here we're looking at anything in the green are microglia proteins enriched in healthy microglia, and anything in the blue are proteins enriched in Alzheimer's disease-associated microglia. And in particular, we see these consistent immune phenotyping proteins where we see these HLA-DR, ApoE, and P2RY12 proteins enriched in healthy, where CD33 and CD44 are enriched in the Alzheimer's disease. And again, this is just visually looking at the healthy associated microglia and some of the more AD-associated microglia showing or reflecting these different functional roles within the general microglia population. So this is less about trying to find a specific microglia populations that maybe interacts with a specific disease protein or whatnot, but just are there general shifts in the functional profiles of these microglia in disease versus healthy, which is what we see here. So the final thing that I wanted to cover is, of course, this is a spatial talk, so we wanted to look at some of the spatial enrichment around these microglia and see what other non-microglia proteins and cell states that they might be interacting with. And again, we used the microglia state continuum to judge to judge what the different non-microglia cell proteins interactions were. And in this case, we narrowed in on a couple interesting little tidbits. And the first here are these different flavors of astrocytes, which is another support protein in the brain. And the more inflammatory phenotypes, was represented by GFAP, were enriched in AD, and the more metabolically healthy astrocytes, as represented by the glutamine synthetase, are shown in the green there. And the final one, which I think is one of the most interesting, are these differences in synaptic proteins. So the one in the green, again, enriched in healthy, those synaptic proteins represent excitatory synapses. So those microglia are essentially more enriched around excitatory synapses, whereas the ones in the blue, those microglia are enriched around inhibitory synapses. So a big problem in Alzheimer's disease is this idea of excitotoxicity, where you just have too much signal running through your brain as your brain starts to break down. We don't really know exactly why that is. However, what we're seeing here are microglia essentially more predominantly found around inhibitory synapses in Alzheimer's disease brain. And it's possible that the breakdown of those inhibitory synapses by microglia could be leading to that excitotoxicity we see. And again, this is just visually confirming that we see these differences between healthy and Alzheimer's disease at the imaging level data. So I just want to quickly summarize. MIBI captures macro and microenvironmental spatial proteomic fine detail in the human brain. Single cell attack sequencing captures a wider variety of microglia phenotypes at a high level, but that ultimately integrating those modalities enables a more thorough mapping of broad and nuanced microglia cell states. And ultimately, I want you to take away from this that the human microglia exist on a phenotypic continuum and that they're not necessarily discrete cell states, which I know as human beings, we want nice buckets, but sometimes we really need to look at the continuous population. These states have different neighborhoods and putative molecular functions, and future work involves looking at how that continuum changes with age, resilience, and potential intervention. So thank you to both the Bendall and Angela Labs, collaborators, and our funding sources. Thank you.
Thank you, Bryan. One quick question. Oh.
So there's a guy by the name of Ian John Deary at the University of Edinburgh who's done a lot of work on intelligence and genetics. And he breaks up intelligence into, and actually Alzheimer's outcome, into basically crystallized intelligence versus, which is kind of memory, versus a form of fluid intelligence. And the genes that he came up with, and what's triggered me to remember this, are a lot of support genes for neuronal function. So I'm wondering, not so much a question, how much your genes for support that you're looking at and found are related in any way to the Alzheimer's outcome versus, let's say, intelligence?
No, it's a really good question. So the single cell attack-seq work that I showed there, that was just on healthy human brains. Whereas with the MIBI samples, we were primarily focused on just those immune profiles at the other end of the spectrum. So I think a big opening point for our future work would be to then go back and be like, okay, all those genes that we saw on the other end of that spectrum, what do they look like in the microglia at the more like functional and proteomic level? And I think that's probably one of the like big next avenues for us.
Yeah, kind of ask the question of whether or not the glial cells are as important to intelligence as the neural connections themselves.
As with a neuroscience background, my like much shorter answer that I want to give a much longer one is the support cells of the brain have been overlooked for so much longer than they really needed to be. I think one of the first things you learn in neuroscience is, oh, your neurons are what causes you to think. You have an action potential. That's it. And then like maybe months later, you'll learn about microglia and astrocytes and but they all also have electrical potential. They all also support the electrical activity in the environment within the brain. There's no reason to think they aren't actively involved in signal processing and what ultimately culminates in thought. So yeah, that's my short answer.
I'm moving you along,
Yep,
Bryan, because
I
we're gonna
know.
keep going.
I
here.
know.
Bryan will be available for further discussion and soapboxing outside after the session today. Our next speaker is thank you, Bryan. Our next speaker is Sizun Jiang, another former Stanford alumni, now stuck over on the East Coast at Harvard. He's going to be telling us a little bit about more spatial atlasing, and what is all? Yeah, exactly. Well, I don't know what to say. I don't know what to say. Well,
I got you.
We're excited. We're excited, Sizun.
Well, thank you, everyone. Thank you, Sean, Mike. I also wanted to let everyone know that this is a very memorable day. It's my second time on the Stanford campus. I'm wearing a shirt. When I finished my PhD, I told my wife, when I do my postdoc, I'm gonna finally dress up. I'll wear a shirt to work every day. I'll wear nice shoes. I did it the one time I came to interview in Gary's office, and I haven't done it since. So thank you for the chance to wear my shirt again. Okay, my name is Sizun, and thank you for listening to me rant. These are my conflicts of interest. And just to give you guys a bit of sense for today's talk, I wanted to give you just a snippet of what we do in the lab. Talk about a couple of stories that we've learned from spatial, just very brief highlights that I think are interesting. We're going to talk about viruses and cancer. So Gary, I just have to keep following you wherever you go. We'll talk about a little bit about spatial multiomics. I do think it's pretty interesting to think about how to do it and different approaches to do it. And finally, some food for thought that I hope will be provocative for the people here. All right, questions at the end. Preaching to the choir here, tissue microenvironment, functional identity, it's great. All right, next slide. What we really do in the lab is really try to combine technology development, technology applications, along with big questions that we are interested in. One pillar of the work in the lab is viral pathogenesis. The next is cancer biology. And the third is immune dysregulation. And I think the really cool projects, and a lot of it has been reflected in all the talks so far and the posters, are ones that you can traverse across the pillars. And even better when you learn something from one of these pillars and you apply it to the other. So that's something I really learned a lot during my postdoc here. Again, this is really where I really hope to be, and we're slowly taking steps in the lab to get there. I realized that the pointer would be helpful here. So one of the things that we recently were thinking about is most of the tools for studying spatial were developed on top of dissociated single cell analysis. And I think that is an intrinsic limitation for how we think about the work, because what it does is we look at things on the single cell expression profile first, right? Some kind of segmentation, feature extraction, you put it into Surat, you cluster with your favorite clustering algorithm. We're missing a lot of components: the single cell neighborhood, the cell types around it, the morphology of the actual tissue, what pathologists look at. All these can matter a lot, I think, to the cell state itself. And so with Zhongming and Gary, along with some really talented students here, we decided we should change that. So the method's called CellSnap. This is really the back end of it. Happy to talk about it in detail after. That's sort of I'm just going to very quickly summarize some of the insights here. Essentially, you can now incorporate all three domains: morphology, neighborhood, single cell features, into some representation, and then do clustering on it. Again, with your favorite tool, Surat, Luvian, it's totally fine. And you can now use all three features to identify things that you couldn't before. So for example, in this case, this is, I think, 1,000-plex cosmics on liver cancer. And we're able to pull out very specific subsets of tumor-associated macrophages that you couldn't before with just features alone. And so, for example, here you can see this in yellow, bunch of macrophages just completely surrounding the tumor over there in gray. And if you perform receptor ligand analysis just to see, are you different? What's different about you? And you can see they completely engage different receptor ligands. And there's really a lot of engagement specifically for macrophages next to the tumor here, and not the ones that were unable to fully infiltrate into the tumor itself. So I think we're now able to use a lot of additional information. And this is just one example of what you can do. There's more in the preprint, and I encourage you guys to take a look at it. The code's on GitHub. The next thing that we were doing for a while too is everyone was trying to do immunotherapeutic research in cancer. And of course, being a new PI, I don't have access to samples. I'm going to do something else. What we end up studying is chronic rhinosinusitis, which is this really incredibly interesting disease. This is done with Jaker here at Stanford, where there are two flavors of CRS. Without nasal polyps and with nasal polyps. Essentially, it's an autoimmune disease. You get inflammation of your airways, really terrible quality of life. Kind of long story short, we uncovered that basal cells have a different differentiation trajectory, whether they come from the epithelium of those without polyps or those with polyps. As you can see here, there's a very clear enrichment at one single decision split point, right, as it's trying to differentiate. It's really where we also see increased IL-4 and IL-13 signaling. And this was rather interesting to us because there is an FDA-approved drug that's an antibody that actively blocks the IL-4, IL-13 receptor that's been approved specifically for CRS with nasal polyps. What it does is within 3 shots, it melts the polyps away. So we managed to get data from Alex Shelik's group that was looking at this, and we were asking enrichment of the genes that are specific for this group over here. And so what they had was single-cell RNA-seq nasal swabs that they did before 3 shots and after 3 shots of the plamotamab, which is this antibody FDA approved, and you see completely depleted the signatures that are associated with that particular transition differentiation state. And similarly, you can do in silico perturbations in which we knocked out the key transcription factor that's postulated to control the split point, and we see this reversal specific for this particular state that's associated with those with nasal polyps. Of course, usually reviewers say, that's cool story. We need a little bit more. So we said, okay, we'll do more. We'll give you 100 patients now. So we did this spatial transcriptomics assay on the genomics where we actually performed single-cell segmentation, annotation of every single cell in there. And we did feature extraction across the epithelium, the immune, and the myeloid. I'm going to cut a long story short. The epithelium is where I'm interested in this case. It's the basal cell trajectory that we are interested in. As you can see, whether it's, basal epithelial cells from the CRS with, without, or control nasal tissues. They do separate a little bit on PC analysis, but what really struck us is when we then sort of put the data back onto the trajectory split itself, the pseudo trajectory, we do see very clear allocation for CRS without nasal polyps, specific for this trajectory here, whereas CRS with nasal polyps is a lot more enriched in this differentiation split here. You can represent it in a different way by looking at transcription scores, and as well as if you look at receptor ligand analysis, it's very interesting. It's almost completely one directional from the epithelial to the immune cells specific for CRS with nasal polyps, which really is what a nasal polyp is. It's a lining of immune cells that form along the epithelium, and when you remove it surgically, it always comes back. So in other words, what there is on the epithelial layer, probably the basal cells, there is some kind of trigger that's forming the microenvironment, allowing tissue remodeling to keep forming some kind of epigenetics memory probably that keeps recruiting these immune cells back, and these nasal polyps keep coming back. And if we can learn how to target that, that might be a better solution than taking shots all the time. Okay, so the next part of the story, I'm gonna divert a little bit and talk about human tumor viruses. We're gonna talk about something that's near and dear to my heart. I did my PhD on EBV. Once you get herpes, it never goes away. So EBV discovered in 1964 by Tony Epstein, as well as Yvonne Barr and Bert Archon. They named it after the first 2, not the third one. Don't ask me why. It's highly associated with a number of different cancers, as well as other diseases: Hodgkin's lymphoma, diffuse large B-cell lymphoma, gastric cancer, and especially in the southern part of China, nasopharyngeal carcinoma, along with more recently, autoimmune diseases, MS, lupus, and for a long time, it's known infectious mononucleosis. What's interesting is 90% of adults are seropositive for EBV, but that doesn't necessarily mean that these cancers will be positive for EBV. So actually only a subset of tumors are positive for EBV. The question is, why are some positive for a tumor virus and some not? For example, in Hodgkin's, 25% or so of the actual cases are positive. When it's positive, it means the tumor cells are positive for the virus. But morphologically, they're indistinguishable, as you can see from this side, right? These tumor cells are these Reed-Sternberg massive multinucleated cells. So we start with a very, very interesting question, and this started when I was in Gary's lab already. What really is different about a virus-positive lymphoma and the virus-negative lymphoma? Is there something we can learn and generalize from that? And so we built a little panel together with Margaret Shipp and Scott Rodick. And I think the results when we saw it was just very striking. It's very rare that when the results come off the machine, you look at it, you're like, wow, completely different. As you can see, in EBV positive, the microenvironment is mainly in purple CD8 T cells, whereas in EBV negatives, predominantly CD4 T cells. This is very reminiscent of the MCC story that Gary was telling you about. Similarly, we also saw that class 1 antigen presentation massively upregulated EBV positive. Class 2, come see, come so. And as well as if you look at T cell dysregulation, again, massively skewed towards memory, exhaustion-like signatures, along with other checkpoint elevated markers. So TLDR from many years of titrating antibodies for this study, T cells are more dysregulated, but what really does it mean? Is there something we can do a little bit more to try to understand more about the T cells really in this case? So what we did was then we asked Richard Burek as well as Scott Rodick for help. We pulled together a much larger cohort. We again used the geomics approach. Some of you might have heard. I'm not going to explain much about it, but essentially it's a 3 antibody stain that lets you do segmentation. We kind of hacked it a little bit in the lab that allowed us to pull 8 different cell types out by doing single cell segmentation, feature extraction, etc. on it. So what we learned, apologies right away, is first we confirmed with this new cohort that the biology is still the same. We saw more CD4s in the EBV negative microenvironment, more CD8s in the EBV positive tumor microenvironment. Great, we didn't break anything. We're comfortable with some of the claims we're gonna say. And then similarly, we can pull the transcriptome from 6 different cell types here. First thing we looked was the tumor marker for Hodgkin's is CD30. The RNA transcript that encodes for CD30 is TNF RSF8. This was kind of an interesting label transfer that was done in the lab manually, but you can see our enrichment for the tumor cells are highly great. Similarly, CD4, CD8, memory naive, most of the markers that we're seeing are quite specific, so that was good. That allowed us to start doing some pretty interesting things. For example, we realized when we looked at the data that the M2 score, the proliferation score for the positive, EBV positive tumors are lower, specifically the tumor itself, than the EBV negative tumors. That's also correlated with having, at least a lot more tumors present in the EBV negative microenvironment. This is from the RNA cohort now. We went back to the protein separate cohort from the MIBI, and we confirmed that similarly, we see proportions for Ki-67 being higher. So in other words, the tumor cell, when it's negative for a virus, seems to proliferate faster. Of course, it could be a combination of intrinsic and extrinsic factors. We first asked, what do we think is going on? So first thing, innate responses. And again, we have virus in the cell right now. Innate responses are rampant within the microenvironment, both in T cells as well as some in the tumor cells. Inflammation is massively upregulated within the tumor when it's EBV positive. So we can see and postulate a lot of the cell's resources is dedicated to keeping a chronic, a virus sort of in check within the tumor cell, and it doesn't replicate as fast, which is quite interesting. Similarly, when you look at the different T cells, we do see more class 1, we see more interferon response, more interferon production, specific for EBV positive. And if you dive a little deeper now into the T cells specifically, we also see more engagement of the MHC class 1 to some extent, at least from the RNA signatures. And this interesting thing where we see cellotoxic up and down regulation, it's a little interesting if you think about it. The gas and the brakes are on at the same time. Thanks to a lot of phenomenal people in the lab, we were reminded that, hey, this is kind of reminiscent of this progenitor terminal T cell state, right? Where, again, like when it gets exhausted, it's not binary, it's on a spectrum, as you were hearing from Bryan just now for even many other cell types too. And particularly for EBV positive microenvironment T cells, they are mostly terminally exhausted. In other words, if you then gave them IO checkpoint blockade like anti-PD-1 that's used right now, they probably wouldn't respond very well because they're at the point of no return at this point. Right, so that's something that was quite interesting that we learned. So in summary, there's EV remodeling of the tumor microenvironment. We're seeing a lot of MHC class 1, CD8, CD4, sort of balances. It seems like it's quite generalizable across many different tumor viruses, we hope, and a lot of immune dysregulations and so on. So this is where I think there are 2 things to be thinking about. The first one is, should we be treating EBV positive and negative? And not just that too, there's so many other tumor viruses. HPV positive and negative head and neck squamous cell carcinoma is treated differently. we should really think about, why are we not doing this the same? It's probably a lot of it we didn't understand, but now we have the tools that let us understand this. But more interestingly, this sort of pushed the lab to think about it. We have this interesting framework. We started first with the protein. And then based on what we learned from the protein, we went into the RNA specifically for those cell types or regions that seem to be, make a difference, at least from the protein analysis. So we're calling this sort of an iterative approach. Now, can we try to do this now on the same slide, on the same cohort, instead of in this study where we did it first in one cohort and we then developed a completely different cohort, just to help us answer the question a little bit better? And to spoiler alert, that certainly is a way to do it. So we're calling this approach in-depth, and it's quite simplistic if you think about it. And I'm a huge proponent coming from a very specific lineage here, that protein matters a lot functionally. But of course, now we learned glycans matter too, so glycans also matter. It matters because you get to do a lot of things. First, you measure the molecular machinery that's doing the work, and also like phenotyping, right? How do you phenotype a CD8 T cell? You use CD8. How do you phenotype a CD8 T cell when you have RNA? Well, good luck with that. Anyhow, so we have this interesting opportunity that you can do very high-plex staining and imaging on spatial proteomics platforms very quickly. The analysis, especially now with a lot of tools being developed by people here, which is amazing, you can highly accelerate the sort of phenotyping or even attention region, elucidation pipeline. So that process, at least within the lab for some of our studies, we can even do it within a day. So what's interesting is, again, measuring the protein may not be enough. And of course, some people, you can measure the RNA first and then you measure the protein. Well, if you measure RNA, you do a protease digestion, there's not much protein epitopes left. That's really what a lot of people don't think about much. I'm sure a lot of you do. While all this is going on, you can then take the slide out, hybridize it with the probes of interest for your platform of interest, and then go for precise region-specific capture. Now that capture can be on a cell type specific level that some of you might be interested in. It might be region ROI specific level. It could be on a single cell level too, depending on which platform and plex you use. And now you have this opportunity to get paired, same slide, RNA and DNA, to do integrative analysis. So that's really what it is. Some of you may not get the reference. I know I'm old. It's okay. But really, there's a bit of motivation for why we're doing this. So again, opinions may vary here. That's my disclaimer. This is meant to be just a very subjective view. Spatial proteomics, you can do a lot of stuff, single section, you can't do RNA, sometimes you can add a little bit, you can do whole slide relatively fast, but sort of, at the limitation of you can't really dissect things very deeply besides some phenotype and functional analysis based on your panels. You're limited to the panel. If you do RNA, there are different methods out there. Generally, I just group them into region capture approaches. You can't really do the protein. Some claim you can, you digest it, you lose some of the protein. Whole slide capability is a bit limited. Ability to dissect is a bit limited because you don't know where you're looking for unless you have to pair H&E, but sometimes the H&E alone isn't sufficient to tell you where to look at. But again, in both cases, the cost and time is relatively linear. You can do single cell, high-plex RNA imaging, can go above 1,000, 6,000, 22,000 soon. Whole slide capability, actually, this is a misnomer. It's actually very unlikely to be whole slide capable, and I'll show you data in a little bit why. But you could, of course, point field of view regions, but the cost and the time is really the limiting factor here. It's going to take a lot of time and a lot of money to do that. You can also do sequential multi-omics, and there are ways, right? You throw the antibodies on at the same time with the RNA, and you pray really, really hard that it's going to be specific. Very expensive, and usually it comes at the cost of sensitivity also. Whereas for in-depth, we're saying, you know what, do the protein first. It's cheap, it's fast, you find out what's different, then you go into the part that you want to go in depth, and then you find something new. Okay, that's where the name came from. Great. But I think, again, every time there's a new method, I wake up at night, right? I'm like, should we really be doing this? Does it really work that robustly? So the lab, thankfully, always tries to reassure me. They developed a robustness test where we took 2 serosections and 1 antibody stained image, 1 no antibody control, goes all the way into probe hybridization, spatial RNA capture, and then we see, do we get back similar levels of RNA, total RNA? And do we get good correlation on the single ROI or single field of view, etc. level? Spoiler alert, first we did codex, you get pretty pictures, then you put it onto an RNA platform like the GeoMix, you get pretty good correlation, you get pretty similar total RNA levels, and your control probes pretty low. And I just want to note that it's a log 1 plus X axis on the Y. You can do the same with manual codecs or automated codecs on the GeoMix or even the CosMix, pretty good correlations, pretty good transcript recovery, pretty, you don't get extra background, essentially is what the control probes is showing. And you can do it automated again for CosMix 6K. You also get similar correlations and also some differences, very minor, sorry. In fact, it's a little bit higher for the protein stain. And so ongoing too, thanks to a lot of collaborators and friends, both here as well as at the Brigham. We also have completed this a couple other platforms, SignalStar, for those of you who want to do high-plex with a commercial CST platform, the Polaris, as well as the Orion here. So I'm just checking the box, Mike and Sean, for your sponsors right here. So really, what does it mean in a nutshell? You do antibody staining, you do imaging, you do rapid phenotyping while the RNA is hybridizing, and you do automated slide capture for each of these cell types that you can phenotype now. And you can learn each of your gene expression programs within the context of each of your field of views, right? So just to show you again, robustness, the protein annotation is pretty robust. The RNA transcription signatures are very specific also. So in other words, we are capturing at least this data is on the GeoMix because it's really fast and cheap. I'm poor, come on. So you get very, very rapid data generation, but also very specific RNA. You can now do fun things because you have the absolute ground truth for deconvolution, absolute ground truth for RNA and protein, and you can test different protein deconvolution approaches. No comments there. Next thing, you can also do fun things such as, in our case here, we were looking at tonsils again to do the benchmarking. We didn't have a TFH marker, or we didn't put PD-1 in there. We just had a very simple hypothesis, which is the more B cells that we see from the protein phenotyping, the more TFHs we should expect, right? But we don't have a marker for the TFH. Because the more B cells, the more it's probably a follicle, then probably the CD4 is a TFH. So then will the RNA give us a proxy for that? And the answer is yes. If you just do a binary split of the high versus the low in terms of how much B cells you have, TFH score is very well split. And also if you do that, just a correlation, the more B cells you have, the more TFH signatures on the RNA you have. Similarly, again, you can find very specific cell type gene expression programs besides just how specific they are for that cell type, but also very specific functional programs out there. Right. So with that in mind, we then decide to go after a question again. There's EBV. I'm sorry, I told you about herpes already. This time we looked at diffuse large B-cell lymphoma. We were able to capture a population, and actually it's a bit more because we then subtype the tumor. I'm not going to go too much into that with the limitation of time. So then you go from multiplex images to phenotype maps to RNA capture. You can analyze some of the features that we saw before. Class 1, class 2 is different, and so on and so forth. What we uncovered is, of course, the T cells are dysregulated. You can really correlate that with effector and exhaustion-like signatures from the RNA side. And this gives us an opportunity to look at the CXCL9 SPP1 polarization that's been described recently, which is great. Again, receptor ligand analysis is now possible. With this case, you can start looking at these different communities that are specific for either the positive or the negative, and then associate that again to a worse or better sort of a outcome. And I'm using this quote unquote because we don't have clinical outcome here. We only know whether they're EBV positive or negative, but the T cell activation and links to macrophages are quite impeccable. You have a lot more activation within the EBV negative and a lot less activation within the EBV positive. So again, now we have multi-omics data. What do we do with the data? And that's usually a question that we get stuck with a lot. And so with Yuzhou and Qing, so Yuzhou's a joint postdoc in the lab with Qing's group, we developed a relatively, I think simplistic but very robust approach that's called spectrograph cross-correlation. Happy to go more into that later at the coffee time, but just to give you a sense, you can start ranking different field of views using codex data, or in this case, any spatial proteomics data. For example, you see global level changes. Your B cells are in the follicle, your T cells are excluded outside, to more local level changes where there's more intermixing because you're no longer looking at a follicle, you're looking at other regions of the tonsil itself. You can do that ranking. You can now ask from the RNA side, what does it look like on a spectrum? And each color here is actually a different technical replicate that we're seeing. And we are seeing quite clearly, and again, the cartoon is summarized that as we traverse across sort of these different combinations of B cells and T cells, their gene expression programs and activated different pathways are different based on the localization and how they intermix to each other. And I think this is really powerful to think about, because that now gives you a chance to very rapidly assess and rank things using spatial proteomics, and then really dive in and look at the RNA pathways related to that. Just a very last minor part, this allows you to plot these pretty interesting pictures that Gary, Salil, and Graham pioneered in their tissue motifs paper. And really it starts to nominate different potential interactions that are possible. You can then nominate out and look at them visually to see that they are completely different in these different interactions. In this case, it's T cells and macrophages again. And what's ongoing now is that we now can do full transcriptome pathway level analysis on these very different programs and pairs, but still at the cell type specific or even combination specific level, because all that RNA information is there though. Okay, so I'm just going to use the last maybe 2 minutes or so to really dive a little bit into the Hitchhiker's Guide for Spatial. By the way, Gary, just want to let you know, I remember everything you told me when I went on the job market. No assay is perfect. Every assay is biased. Some assays are more biased than others. Knowing your platform strengths and weaknesses is the key to success. Rubbish in, rubbish out. If you're going to do spatial, you need to understand histology and pathology, and I believe some basic biochemistry too. Sean, I really agree with you on that. And this is why, right? First thing, tissue processing really matters. This is data that's we've been wanting to do this for a long time, so we're just so glad now to be able to do this. How do you decide on what's the epitope retrieval buffer and time and temperature for your study? You probably go to like the last paper, like Gary's cell paper, and be like, oh yeah, 97 degrees, 20 minutes. I'm going to do that. That's what we did. But how did we come to that? And is it the best condition? So then we've now proposed to systematically go after that. And in this pilot data, initially, we only went after 4 conditions, right? 20-minute, 10-minute, or 40-minute retrieval at 97 degrees in a very specific buffer, the same buffer for all of them, in the same instrument, done at the same time. Antibodies stained with the same cocktail, lot controlled across different temperatures, 37 degrees 1 hour, 4 degrees overnight. You then do the data acquisition. Right off the bat, you can see already by eye, your images are looking different in terms of how intense they are. These are the raw data, actually. If you then do your segmentation, feature extraction, and plotting of all the markers out there, completely different. And Revere had a really good suggestion. So Cancun and Team's lab then plotted also the variance of each marker. And we can now then systematically rank what is probably the best condition by sort of looking across all the different markers. And for example, this condition, which is 20 minutes, 97 degrees, 4 degrees overnight staining, has consistently the top best staining conditions in terms of signal to noise. Okay, so now we're really expanding this up to 24 conditions at the same time, validated across a number of different platforms. Thank you. A lot of you are in the room who are supporting this together. So we really hope that this will be something for the community that we can say, systematically evaluate what exactly are some of the retrieval and staining conditions. And honestly, this is what it is, right? Because retrieval and staining are just reverse cross-linking and KD of antibodies. Next thing, it also matters for RNA. So this is a preprint from Sammy's group that really pushed us a little bit to think about it. So what was done was they then systematically went through exactly what the manufacturer recommends for spatial transcriptomics. And what you can see is that the sectioning storage is different, the tissue baking is different, the retrieval buffers and times are completely different, the protease digestion, which increases or decreases accessibility of the probes, are also different. There's a lot of things which are different, but what we were doing in the lab at the same time, actually, and this was actually for an internal study, we didn't want to use the study for this, is if you take 2 sections of a tissue microarray and you process them exactly in parallel with a retrieval buffer that's slightly different, you get 50% difference already in total genes and total transcript. That alone gives you a massive improvement in cell type annotation. And another one that we did, again, just throwing things out there, we had a clinical trial cohort that we decided we are going to test 2 conditions of tissue processing on the cosmics. And you can see the results are about 20–30% difference in amount of transcripts we get back. And we said, you know what, this looks great. Let's take condition 1 and let's run it across the entire cohort. So that's what we did. We ran it across the entire cohort. And this is what we got back. Now the question is why? And I think we learned 2 things from this. The first one, if you notice, this is a tissue microarray. Things are in fours. Your total transcript travels in fours here. Your total transcript travels in pairs here. Tissue quality, number 1 determinant is the source of the tissue because fixation is not standardized, right? How long was it left out, etc. So many other conditions. But the next thing is we imaged about 40 or so field of views here and 100 here. The time that we used to image was 3.8 days versus 2.5 days. Time alone gave you a massive difference in sensitivity of the probes. Right, so I'm going to update this a little bit, Gary. I'm going to also add reverse cross-linking. It's pretty important to think about. KD of antibodies is pretty important to think about. And K off of RNA probes as a function of time is pretty important to think about too. So with that, I get the hint. Thank you.
Thank you, Sizun. Now we know why you can't afford to do the atlases because you're too busy doing everything else. So I can take maybe one quick question. Is there a question online? If not, I can.
Oh no, Gary's got a question.
Oh.
Specific question. Thanks for a great talk. Which pH for antigen retrieval is preferred?
We don't know yet. We showed 9 in this case, but the benchmarking will show citroconic anhydride too. Yeah. All right.
All right. Thank you, Sizun. So I've got a couple quick closing remarks, but thank you everyone again for coming. There's going to be bagged lunches for people to take away outside as soon as this is over. And how's it going here, Frankie? Am I still freestyling? I'm still freestyling. And I think I'll preface it now. The very last slide I'm going to leave up is going to be a QR code for a survey link. If you miss it here, we'll also be emailing everyone who registered. Hopefully all of you actually registered and there's no gate crashers here, but we're always looking for more feedback for the meeting. We're thinking about what this is going to turn into next year. Are we going to do it again here? Are we maybe going to go east? Are we going to, I don't know. We're this started really as honestly, a proteomic imaging workshop 3 years ago. And we had a whole bunch of people showed up who didn't have these platforms, who weren't actually using these tools because they were just interested in what to do with these data sets. And so it made us realize that there's a community around, what you can do with these things. I think, again, just to show this slide that Mike showed at the very beginning, we had almost 300 people between live and online stuff registered for the meeting here, covering multiple countries and academic institutions. I think it's a testament of how many people are still here at the end of 3 days of spatial biology talking. I just wanted to kind of revisit something that came up yesterday. And a few of you have offline talked to me about this, where a lot of our colleagues grumble about why does spatial biology even matter? And so I just wanted to kind of point out a few themes that have come up because I think it's important. It's not just another way to do single cell analysis, right? I think broadly, we know that cell identity and function is an intrinsic product of its environment. I think that's a theme that's come up in a lot of talks. The cells, we can only understand what a cell is based on where it is and its neighbors. A lot of time that dictates the cell's function. I think, a lot of the talks, particularly a lot of the talks this morning on this last day, showed in disease, especially with tissue dysfunction, things like tumorigenesis, structural biology is strongly predictive and associated with what's going on. You can't tell what's going on just by cell composition alone. And I think a lot of the AI talks yesterday emphasized how much of pathology and diagnostic medicine is uniquely structural and spatial. So, that's not going away. And really from a practical sense, I think the other cool thing we saw with a lot of the data analysis and data digesting tools is that our cellular data sets are better when they're derived from images instead of just deriving out of a single cell data point in silico where you can't actually see the information, right? And often too, from a practical sense, tissue is sometimes the only sample, right? Like sometimes we just can't get nice single cell or disassociated data sets, right? So sometimes you have to be able to go back to tissue and go back to the archive. In terms of outstanding challenges, you know, I think this is, Sizun went through a little bit of this with his Hitchhiker's Guide, but I mean, I just want to reiterate, I think the thing we tried to emphasize here is there is no one omics to rule them all. This is not Lord of the Omics, right? This is, I think we're all using awesome tools and we're all asking similar questions in different ways, but we know that any one tool can't capture all the information. That I think the other big theme, and it was really nice with Emma having a recap at the end there, is that there is a lot of computational challenges, right? So we can generate data that we still can't fully digest and interpret. And so there's, I think we're all going to be employed for a while, fortunately, to do this. And it really touches on a theme, actually, that I don't know if anyone here knows Mark Davis, but he's kind of one of the granddaddies of immunology here at Stanford. And he gave a talk at our retreat a while back. And one of the things he brought up is that like the scientific method is all about iteration, right? Like we don't do an experiment once and discover. We often discover and make science better by doing things over and over again and repeating them. And so with spatial biology and all these big data issues, it's only worse, right? Like we spend all this time looking at, you know, these precious data sets we've collected. And so I think in terms of how expensive and how challenging this can be, this is still ripe for disruption so that we can iterate and do things faster and, you know, again, improve science around it. So, you know, and I guess also from another standpoint is we've already kind of gotten to the point in this like technology race where you can't just go and run a Xenium or run a Codex or run a whatever and get your Cell Science or Nature paper. Like the editors have figured it out. They're asking for more experiments. They're asking for stuff. And so I just want to hit on the fact that, again, we saw this in a lot of the talks. This is just information and data. And it really is, I think, to really the next bar and to show it matters, to really demonstrate to our peers that spatial biology matters, is we have to figure out how to turn it into knowledge and wisdom, right? We need to perturb the system. We need to show the system's testable. We need to show that we can expand what we know about stuff. So that's the challenge for future years. Thanks again. This would not have been possible without money from HubMap and the NIH and our sponsors and you guys being willing to pay some registration fees. So that was awesome. Thanks again to the team that really inspired the generation of the program and chased all our excellent speakers around. I also wanted to give a shout out. This is our eye in the sky. Meena Shivan is also online. She's actually a coordinator on a number of our consortium grants, and Meena actually did a lot of the background work to make sure that the financial accounts were set up and that all the room booking and organization stuff was done. So it was fantastic. And again, as promised, please give us feedback, let us know how we're doing, and give us some suggestions for future meetings. Thank you again.